And now we move on to our next presenter, the most awaited presenter, I would say, Dr. Raghunath Swaminathan, a PhD, a molecular biologist, a scientific advisor at Alpha Naturals LC. Sir is going to speak the most important sessions today. He is going to address on detoxification process inside a cell during fasting. He will address on intracellular metabolic pathway during infection and inflammation. He will also mention the importance of tridosha and the molecular basis of it. Dr. Swaminathan is a molecular biologist. Today, he will explain how detoxification processes activate inside the cell. He will focus on the mTOR pathway dependent autophagy activation. When this intracellular cleansing process is inhibited, the accumulated non-functional cellular components become the fundamental cause of inflammation, cancer, and other neurodegenerative diseases. The molecular changes that occur during the development of autophagy inhibition, he will cover the entire topic on how detoxification process occurs while we fast. So, Dr. Raghuram Swaminathan, it's an honor to have you with us. May I kindly request you now to do your presentation.
Yeah, thank you so much. So, am I audible?
Yes, sir.
Yeah, I've shared the slide.
Yes, sir. Kindly switch to a PowerPoint mode or a slide mode.
Yeah, I have shared my slide. Maybe I'll try one more time.
We could see your screen. Just have to.
Yeah, it's coming. It's coming.
Yeah, kindly switch to the PowerPoint mode, sir.
Yeah. So, can you see my slide now? It's loading.
It's loading.
Is it okay now?
Yes, sir. Yes, sir.
Okay, great. So, thank you so much for this opportunity. I have 3 stories to tell you today. The first is about the detoxification process upregulated inside your cell during fasting. The second is about viral infection and cytokine storm and lung fibrosis. So when there is high calorie diet, high glucose and high protein in your blood, how this is going to increase the cytokine storm, because glucose and amino acids are required to make cytokine secretion happen. So how by controlling glucose and amino acids we can control cytokine storm and lung fibrosis and fatality during a viral infection by controlling a pathway called hexosamine biosynthesis pathway. So that's the second part of my talk. Third part of my talk, I will focus on how from the millions of plants around us, how the Ayurvedic physicians or ancient herbal medicine practices and sages understood that which herb should be used to cure one particular disease. So this was always our curiosity, and so let's go to our first story.
Dr. Swaminathan, there's some sound disturbances from your end. I think it's the wire or your mic. Maybe.
Okay. Sorry. I'll remove. Is it okay right now?
Yes, sir. Very close.
Yeah. Nice. Okay. So first thing is about autophagy. So as Dr. Rao already explained about this, but I will show you the confocal images, microscope images, and Western blots, and all those laboratory experiments which led Professor Yoshinori Osumi's group to understand autophagy and how they understood its role in diseases. So let's understand autophagy in basic level. What you see is a plasma membrane of a cell. This is inside of a cell. This is outside of the cell where the blood flows. So when you are fasting, there is very less amino acids and very less insulin. So these concentration of amino acid and insulin are sensed by an enzyme called mTOR, or target of rapamycin. So this enzyme is a master regulator enzyme. So this always catch the autophagy enzymes and inhibit the autophagy enzymes. But when amino acids or insulin are less in the blood, the mTOR is suppressed. It becomes inactive. Then the autophagy enzyme run away, escape from mTOR, and go to another portion in a cell where it will start making a membrane which will look like a basket. So this is similar to plasma membrane, and this will look like a basket, and it will keep on enlarging, and then it will engulf all the toxic materials inside the cell. For example, mitochondria. Mitochondria is the powerhouse of the cell, and it needs to make energy molecules called ATP. It needs to make amino acids. It needs to make fat metabolism. So after working for prolonged period of time, mitochondria gets damaged. Then it starts making reactive oxygen species or single oxygen, which are highly reactive. It can hit the DNA and cause DNA mutation and can cause cancer. Then there are proteins inside the cell, enzymes, which after working long hours, they will become damaged, old, and these are very toxic. They are hydrophobic, they can repel water, then this can cause neurodegenerative diseases. So autophagosome, which is this organelle, autophagy is the whole process, and so autophagosome will engulf all these and finally fuse with the acidic compartment full of digestive enzymes. So this lysosome has the caliber to chop up everything which the autophagosome brings, and so finally they fuse together, fuse and chop up everything, all the toxic materials, and it's all recycled back to the cell like basic components like amino acids, glucose, nucleotides, which can be used to make DNA. So it's a recycling process. This is similar to when you have a weekend and you don't need to work, then you would engage in removing the decayed components in your house. Like you will want to clean up your fridge, which has a lot of old food, which is causing some bacterial fungal growth. So similarly, and we can also tell it as a story of a township. If a town is under lockdown for 1 year, we cannot get any more components from outside. No raw materials will come from outside. So we won't get bricks, we won't get cement, we won't get iron rod or wood. Then what the town does, like if our family will be expanding, we need to make another house, we will decide to recycle the old non-functional offices and houses. Old houses, we'll take the bricks and wood and iron and pillars, then we'll make a new house. And this is what autophagy does, it recycles. So in this process of recycling, you will basically end up cleaning up all the toxic components inside the cell. Second part of my talk is about virus infection and what happens inside an immune cell during virus infection. So in a normal cell, this is where the blood flows, this is inside the cell. So there is glucose entering the cell, and this glucose is converted to pyruvate, and this pyruvate enter the mitochondria, and it will make 36 energy molecules called ATP. However, when there is a virus infection or cancer, the cell lost his inner planning. So when the glucose enter the cell, there's a pathologic condition where this glucose is cut into two pieces to form pyruvate, but it cannot enter mitochondria. Instead, it will be converted into lactic acid, and there will be an acidic atmosphere all over here, and it will make only 2 energy molecules or 2 ATP. But the cell requires 36 ATP. So what to do? This cell will uptake tenfold higher glucose. So then there is a Warburg shift. This was discovered by Otto Warburg, and this is probably the reason that when you are fasting, the tumor cells die because tumor cells require tenfold higher glucose than the usual cells. Because tumor cells cannot make 36 ATP from one glucose molecule, it requires 10 times more glucose. But when you're fasting or when you're in a caloric restricted diet, you don't get tenfold high glucose. So the tumor cells will eventually start dying. Okay, but that's not autophagy, that's about tumor cell death. Now, once it was noticed that in diabetic patients there is hyperglycemia, there's high glucose in the blood, and they were having lot of cytokine secretion and cytokine storm. So what are these cytokines? Cytokines are the protein, hormone like proteins, they are not hormones, proteins secreted by immune cells. So immune cells language of communication is cytokine. Once cytokine is secreted, immune cells will ask dilation of blood vessels. Then millions of immune cells will come to the site of infection. Then it will ask the other immune cells to make antibodies. But if the cytokine secretion gets abnormal, it's get abnormally high, what you see in virus infection fatality. The lungs get filled with fluid and there will be a complete traffic jam of immune cells start accumulating. Then there is lack of oxygen in the lungs and there is lung fibrosis and eventually it ends in fatality. So in diabetic patients, it was noticed that there is cytokine storm. And when they studied it in detail, they understood that in diabetic patients, when glucose and amino acid enter the cell, so this is a plasma membrane, glucose and amino acids are entering the cell. Now, this is a traditional pathway. It will be broken down into pyruvate. Pyruvate will enter mitochondria and make 36 ATP, but it's not going to happen. It will stop with pyruvate. It will be converted to lactic acid, as I said before. And there is a tenfold increase in glucose here. And this glucose will be rediverted to another pathway called hexosamine biosynthesis pathway to make cytokines. So that's what you see here. This glucose, instead of going to pyruvate, it will be redirected to become another pathway called hexosamine biosynthesis pathway, where it will make amino sugars, means combining glucose and amino acid. Sugars, amino sugars, which is required for cytokine production, cytokine secretion. So the interesting thing about is when there is a virus infection or cancer, there is a tenfold increase in glucose uptake. And this is not a normal physiology, it's a pathologic condition of the cell. And out of this tenfold increased glucose, 5% of the glucose will be rediverted to hexosamine biosynthesis pathway to make cytokines. And when there is high caloric diet during an infection, you will be producing more cytokines because immune system is not connected to brain. So brain cannot tell immune system directly that stop this much cytokine. It is going to kill the human being. It cannot tell directly. Immune system responds to the food. So when you catch cold, you might realize that you are losing taste and you are losing hunger. You don't feel taste when you eat some tasty food. So brain is trying to tell you, brain is demanding that you should not eat food when you catch cold during a virus infection, so that the glucose level can be reduced and cytokine secretion can be controlled, because neurons from the brain cannot directly connect to the running around immune system. It can only communicate through your mind to the immune system. So if you don't take food, the glucose will be less, immune system will produce less cytokines. But so this is what, when you are under a low calorie diet or when you're fasting, the 5% glucose diverted for hexosamine biosynthesis pathway will be reduced to 0.5% or 0.1%. So glucose and amino acid is required for making cytokines, but eventually when the glucose is reduced and the hexosamine pathway is only 0.5% functional, you will eventually have a very low cytokine secretion, only necessary for killing the virus, not a hyperactive immune system. So we will cover details of this in the second part of my talk. But anyway, the third part of my talk is that there are millions of herbs in the mountains of Himalayas, or let's say anywhere in the world. And traditionally, the herbal medicine practitioners and sages understood which herb can cure which particular disease. Like you have prostrate, you have a particular herb to cure that disease. You have diabetes, you have a particular herb to create that disease. You have a depression, you have a particular herb to cure that depression. So every disease has a special classification of herb, but there are millions of plants around. How did they recognize this? So there is a very intense biology behind this. So let us try to understand. There is a vast similarity between Aristotle's biology and fundamental cause of a disease development, which Aristotle has described, with what Ayurveda has described about the fundamental cause of disease formation. They come to the same similar background, that imbalance in the five elements: air, water, earth, fire, and space. But it's not that simple as we think. It's not just air, water, and earth. It's a very intense concept beyond the understanding of our brain. But let us try to understand this tip of an iceberg. So space plus air is vada, or we can say its characteristics as dryness and cold. Like you are not going to sleep for like 3 days, your body will experience dryness in molecular level. It's not the dryness what you feel, but dryness in cellular molecular level. And you might experience cold in your feet. So this is something really happening, but we do not know what it is. Now, earth and water combined to form Kaba. This is not actually the cold, but it's more advanced explanation. But if you catch cold and you are going to drink sweet sugar, milk, and biscuits, you are definitely going to increase your lung infection with cold. So, what is the fundamentals of this? Taste of a plants. They extensively classified the taste of the plants, and they found that one particular taste can balance one particular imbalance. For example, if there is a bitter taste can balance or reduce kaba. So bitter taste can reduce the fundamental cause of a cold and a viral lung infection. But if you take sweet taste like milk, glucose, Oreo shake, or biscuits during cold, your kaba dosha will increase. Or you can say that the fundamental cause of your cold is going to increase. So let us try to understand the molecular base of this. So am I audible now? Can you follow what I'm trying to say?
Yes, sir.
Yes, sir, you're audible.
Yeah. So immune system is evolved to understand these tastes. This is the interesting part of evolution. The bacteria, when they infect, they secrete some compounds which are actually bitter. So whenever there is a bitter compound, immune system think that there is an emergency. So immune system will declare an emergency in the body. So whenever there is secretion of bacterial products, it's bitter. This is the lung epithelial cell which has sensory chemosensory cells. So this bitter compound will bind to T2R receptor and it will transmit signals. Then the calcium outburst happens from endoplasmic reticulum. Then this calcium outburst, calcium is a signaling molecule. It can transfer from one cell to another and it can just spread everywhere in the lung. Then calcium will promote secretion of antimicrobial peptides and antiviral peptides from the lung cells. So whenever there is a bitter taste coming up, immune system think this is an infection and it will start secreting antibacterial and antiviral peptides. This will cause holes in the bacteria. It will kill the bacteria. It will destroy the virus. And let us think the interesting part. According to Ayurvedic theories, if you take sweet taste, it's going to increase the symptoms of cold. Not symptom, but the fundamental cause of cold. So how this happen? If you take glucose, glucose will bind to this another receptor called T1R receptor. This is going to inhibit calcium release. So calcium is not released, antimicrobial peptides are not secreted, antiviral peptides are not secreted. You are going to work against your own immune system when you are taking high caloric diet. So what my students do is whenever they catch cold, they cannot have a nice taste. They cannot enjoy the food. So they start taking very tasty food like ice cream and chicken fry and Oreo shakes. It's a cream biscuit plus ice cream shake. Can you imagine? Because they cannot enjoy taste. So next 3 4 days they start taking this very sweet things which the new generation do, and then you have a sleeplessness which will increase your dryness in your body, which will further increase. It will rupture the mucosal system, damage your lungs, and this is creating a havoc. I will come to details of this in my third part of the talk. So all the experiments done till in whatever I am showing is done in mice and monkeys and in cell culture studies. So there is no evidence it's going to work in human. So do not use this for any kind of treatment purpose. It's not. It's only for your own knowledge. It will open up a research mind inside you, and you will be able to view the disease and your patients in a very different angle. So before going to all these, I will start with basic cell biology because we have a wide range of audience starting from undergraduate students. So this is a mammalian cell. What you see is a plasma membrane, the outer border, and here is the nucleus where DNA is and the decision-making brain of the cell. And here is the powerhouse of the cell, which is mitochondria. These are railroads where the mitochondria can catch up and move to one to another place. And this is a endoplasmic reticulum where DNA asks the amino acids and ribosomes to make a enzyme or protein like insulin, for example. When there is high glucose, there will be more synthesis of insulin from endoplasmic reticulum, which will be exported to Golgi complex and it will be secreted out. There are lysosomes, which I already said, they are acidic compartments full of very powerful enzymes which have the capability to degrade anything inside the cell. All the byproducts or damaged products, it can recycle. So this can be compared to a town, the town planning. Like we have the policymakers, the chief minister and the ministers who will decide what to do inside the town. There is an electricity board which can make power or the current, the electricity. There are roads and rails which are microtubules where the enzymes and proteins can travel, and there are bus and car like stuffs called motor proteins which can take these mitochondria to another place, which can take autophagosomes to one place to another place. So there are railroads in the cell. Then there is a skeleton inside the cell which keeps the cell in this position. Cells also have kind of legs by which they can move around. So it can also divide. So this is something like a town. And there is like, let's say, corporation of Cochin, where you can degrade or remove all the waste products in the cell and dump it in this place, and everything will be put to fire and everything will be recycled back to the cell. It will be reused. So this is a starch molecule and this is a glucose. So starch is made from simple units of glucose. Glucose has carbon chain forming a ring and several glucose coming together to form a starch. Now, this is a fat. So you might realize everything is a carbon chain made by the plants, which we get. So this is a fat structure made of carbon chain. This area is hydrophobic, which means it cannot dissolve in water. That's why oil is floating. So this is a cholesterol structure, again carbon rings. All these are entering your body and your cell is trying to process it. This is a basic structure of amino acid which makes proteins. So amino acid has again carbon, carbon, then there is a carboxyl group and an amino group. Now these amino acids will join together. There are different types of amino acids and they will join together to form a protein. So based on the type of amino acid, you see each color represents a different amino acid. Depending on the combination of amino acid, it determines what function the protein will do. Like I said, some proteins work like motor proteins, like they are working like a transporting system inside the cell. Some proteins work like a turbine in the electricity board or dam, which makes electricity. So in mitochondria, there is a turbine-like protein which can keep rotating and make energy from glucose. You cannot make energy, you can convert one form of energy to another form of energy. So there are different type of proteins. So this is a cell and this is nucleus, and nucleus has DNA. DNA has codons, several codes, and each code will code for one particular amino acid. So DNA makes RNA. From RNA, there is a code. Based on the code, different amino acids join together in the ribosome machinery. So what ribosome does is it will bring amino acid and it will rewrite the code of RNA and it will form a protein amino acid chain, which is a protein. Now, there is another mechanism which happens is cell signaling pathway. What happens is this is where the blood flows and this is inside the cell and this is the nucleus. When there is some signal from outside, let's say insulin, it will bind to a protein receptor on the cell membrane and this signal will be transmitted downstream. So just like from the brain, neuron transmits signals to our leg and brain asks us to lift the leg and we can lift the leg. But cells do not have neurons, but they have signaling pathways which can transmit the signal and finally it will reach the nucleus. Then RNA will be made and from that protein will be made, let's say insulin. It will be secreted out to manage the glucose in the blood. Let's contain another situation when there is a bacterial infection. Bacteria has lipopolysaccharides. So these lipopolysaccharides bind to a receptor on the cell membrane and this will activate a protein below this and it will finally activate a very important enzyme NF-kappa B. And this will be transmitted inside the nucleus where it will make the cytokines. And cytokines will be secreted out from the immune cells when there is a bacterial or viral infection. What does cytokines do? They are the communicating language of the immune system. So cytokines will dilate the blood vessels, then it will recruit millions of immune cells to the site of infection. It will ask the immune cells to destroy the infected virus and it will ask other immune cells to make antibodies. And when cytokines become abnormally secreted, it will cause fatality. It causes lung fibrosis, fluid accumulation in the lungs, then finally the patient die. So I will come to the first part of my talk, that is autophagy. when there is nutrient abundance, you are having a high caloric diet. You have enormous amount of amino acids and glucose in the blood, and there will be insulin and growth factors. So cell understands that there is lot of food, it's time to grow and multiply. So there is a master regulating enzyme called mTOR. What mTOR does is it can receive the signals from glucose and amino acids and growth factors, and mTOR will ask the nucleus to make more DNA. It will ask the ribosomes to make more ribosomes so that more protein can be made. Ribosomes make the proteins. It will ask the DNA to make more proteins. It will ask, so you need to understand proteins form enzymes, and enzymes regulate the cell. Like if the cell want to divide, it's done by the enzymes activity. And it asks the lipid to make more lipid synthesis. Why is that? Because cell wants to divide, make a copy of itself, and it can actually divide 48 times or more. But mTOR will also inhibit autophagy, because when it can be assumed that same as our life, when we are busy with our job, we have lot of money, we will think of making another house and expanding our family. So we will try to save money. So food will be stored in the cell, and it will think of growth, and we won't be cleaning up. But when we are fasting, all this process is stopped, because mTOR is suppressed when there is no amino acid and no insulin. So suppression of mTOR will make ATG protein, which is the autophagy enzyme. It was inhibited by mTOR, but now that mTOR is suppressed, autophagy enzyme will run away from mTOR. It will escape and it will go to another portion of the cell where endoplasmic reticulum is, and then it will make a lipid membrane which looks like a basket. And now this is going to engulf the abnormal mitochondria, which is toxic for the cell, which makes reactive oxygen species. It will clean up the old proteins, and finally it is going to fuse with lysosome. Lysosome is full of acidic enzymes, and everything will be chopped up and recycled back as basic components of lipid and amino acid, glucose, and nucleotides. So nucleotides will be used to make DNA. So what is it? Mitochondria is made of lipid complex. Like plasma membrane, I showed you the basic structure of lipid. It has carbon-carbon chain, but billions of carbon-carbon chain form a lipid membrane. And millions of amino acids form different type of proteins. Millions of nucleotides form DNA. So all these are basic bricks which make a big building. So this will be broken down into basic bricks and given back to the cell, so cell can recycle and use for the new generation of DNA, protein, and lipid. So this is again the same thing I am trying to repeat. Low glucose and low amino acid inhibit mTOR, and this will increase the lipid membrane, which forms like a basket. It enlarges, it will engulf, damage mitochondria, damage proteins. It will take, if there is a virus running around or a bacteria running around, it's going to capture that, and everything will be destroyed in the lysosome, burned and recycled. So this is how a cell will look like in microscope. One of the early experiments from Yoshinori Osumi's lab. So what they did was they fused the fluorescent proteins you see in coral reefs. They fluoresce in the night, right? So they take this particular DNA, which makes the fluorescent proteins. Then they will fuse with the autophagosome membrane protein. So eventually you can see them fluorescing inside the cell. So in a fed condition where when you are taking food, autophagy happens in a very minimal level. As you can see, tiny dots, When you look under electron microscope, they are like a basket engulfing some unwanted proteins. And when you are fasting, there is less amino acid, less serum, less amino acid, less serum, and less glucose. Then you will have a tenfold increase in autophagy. So tenfold increase means there are like hundreds of autophagosome running around in the cell, so busy cleaning up the cell. And here you can see that they are cleaning up many accumulated machinery inside the cell. So this is a autophagosome. It's a double membrane structure, very stable enough to take up any toxic component. So this is a healthy mitochondria, elongated. You can see a stable cristae where energy is prepared as ATP. And this is a damaged mitochondria. You can see they are fragmented and round instead of elongated. So these mitochondria are toxic for the cell. They will make reactive oxygen species. Then all these kind of mitochondria will be engulfed inside an autophagosome, and it will be eventually degraded in the lysosome. Similarly, you can see that this is an autophagosome engulfing lot of damaged, misfolded, toxic, hydrophobic proteins from the neurons. So damaged mitochondria will make reactive oxygen species which can cause cancer and death. And damaged misfolded proteins are hydrophobic. They can bind to anything in a cell. So they can cause neurodegeneration. So there are different types of autophagy. One is a general autophagy, which will generally take up anything. Another is a specific autophagy, like you call it xenophagy, where if there is a bacteria running around inside the cell, it will be specifically recognized by an autophagosome and eat up, and it will be burned and destroyed inside the lysosome. Similarly, if there is a damaged mitochondria, it will be creating a lot of reactive oxygen species or reactive oxygen atoms, and this is dangerous for the cell. It can cause DNA mutation and cancer. So it will be specifically taken up by autophagosome, calling it a mitophagy. And ubiquitinated proteins are misfolded old proteins, which again will be specifically engulfed. So what happens if autophagy is impaired? Damaged mitochondria accumulate. So there are hundreds of mitochondria in a cell, and you imagine hundreds of damaged mitochondria accumulating in a cell. It will be like you have many old factories in your town emitting heavy toxic fumes, and you have all the vehicles emitting fumes of bad oil, diesel and petrol. It's full of brown, black color pollution happening. Definitely you're going to become sick. And you have bacteria and virus running around. So when autophagy is inhibited, all these slowly start increasing. And this will activate NF-kappa B pathway, and it can cause inflammation, and if not controlled, it will cause apoptosis or cell death. So let us try to understand autophagy. We need to inhibit autophagy. Only then we can understand what is the importance of autophagy, because when we are living a normal life, everything look normal to us until after 40s we start getting diseases, after 50s. So when we inhibit autophagy, what happens? That will show the importance of autophagy. So in mice, when autophagy was inhibited in a laboratory conditions, this is a mouse liver, and you can see that as it becomes 19 months, this liver is full of tumors because autophagy is inhibited. And when they took sections of these organ, they found that, so this is a healthy mitochondria from a healthy mice. Electron microscope images show a beautiful elongated mitochondria with cristae inside, very stable, and which will be making ATP from here, 36 ATP if you remember. But when autophagy is inhibited, this is one cell under electron microscope. This is a nucleus, and all these round you see are damaged mitochondria. They are not elongated like this. And if you see the zoomed version, they do not have this cristae. This cristae has the turbines of the electricity making turbines. I said there are proteins which act like turbine. They keep rotating and make ATP from a charge potential gradient. And this is where it is located, the turbines. But this whole crystal is damaged and you don't see it anymore. So all this damaged mitochondria, they cannot make 36 ATP. So the glucose coming inside will be converted to lactic acid, and there's a high acidic atmosphere in the inside the cell and outside the cell. And this acidity itself will activate cytokines. But anyway, so this is what happens, and there is high oxidative stress definitely, because lot of singlet oxygens which are very unstable. Unstable means they have electron less or an electron more in their outer orbital. They can be very reactive. They will hit anything like DNA and it will cause a mutation, and that can cause cancer. So why would a DNA mutation cause cancer? Is that there are enzymes in the cell which tell the cell that it should divide only 48 times, like Hayflick's limit. So there is a inner consciousness of a cell, but this enzyme tell the cell how many times it should divide, when it should divide. But when that enzyme get mutated because of DNA mutation, the enzyme lose its self-consciousness. So the cell will start randomly dividing. Anyway, so this is the normal mouse brain. And you can see that tunnel staining specifically stains dead cells, neurons. So you don't see much staining of tunnels here, but when autophagy is inhibited, the mouse brain, you can see that lot of tunnel staining dots. So all these are dead neurons. So when autophagy is inhibited, neurons are dying and it causes neurodegeneration. And when you analyze the sections in detail of the neurons, so this is a healthy mouse brain, you see that the neurons are okay in a histochemistry. And when autophagy is inhibited, you see lot of misfolded proteins are accumulating in the neurons. So much. And this is very hydrophobic and toxic for the neurons, and definitely neurons are going to die. And if you extract the brain and run it on a gel, which is an SDS gel or Western blotting, this is a technique where you can detect proteins. So this band will be formed of like millions of proteins forming a particular size, so you can see in one band. Here you see a lot of ubiquitinated proteins are old misfolded proteins which will be tagged by another protein called ubiquitin. So they are at a higher molecular weight, they appear staggered. So a lot of abnormal proteins accumulating when autophagy is inhibited. So I'll conclude a small portion of autophagy, that is part 1, where I explained that mTOR is a master enzyme which regulate the nutrient abundance, and it decides that the cell should divide or not, what the cell should function, growth activities. When there is fasting, mTOR is inhibited because there is no more glucose, amino acids, and insulin. And mTOR inhibition causes the escape of autophagy enzymes, and autophagy enzyme will upregulate autophagy. Autophagosomes will remove toxic machineries like damaged mitochondria and damaged proteins. Then inhibition of autophagy causes tumors and neurodegeneration. Autophagy also plays a very important role in fighting viral infection and inflammation. So far it's getting clear?
Yes,
Yes,
sir.
sir. It's very clear.
Yeah, okay, thank you. So there is some more to finish autophagy, maybe 5-10 minutes more. So this is not autophagy, this is a macrophage, and I am trying to explain phagocytosis. So here is a bacterium or virus, and once it enter inside the cell, it will be eaten up by the macrophages. When there is virus in the lungs, it will be eaten up by the macrophages. Then this virus is inside phagosome, which is again a lipid membrane. This is not autophagy, okay? Then this phagosome will fuse with lysosome, which is full of digestive enzymes, acidic compartment, and the bacteria or virus will be burned to death. It will be chopped up and thrown away. Very heavy defense. And anybody who escape this phagosome and run out, they will be caught by autophagosome. So if a bacteria is running away from the phagosome, autophagosome will run behind it and catch it, and it will fuse with lysosome and degrade it. So this is a recap of the whole story. When a bacteria or virus enter, it will be eaten up by phagosome, which is a lipid compartment which looks like a ball. It will fuse with acidic compartment lysosome, and everything will be burned and destroyed. But some of the bacteria and virus break this lipid membrane and escape. They will be shot dead again by autophagosome. Autophagosome will catch it and engulf it and degrade in lysosome. So there is 2-3 line of defense inside the cell itself. Similarly, virus end up in a similar fate. And it's a paper published in Cell, very important journal in molecular biology field, and the top journals in molecular biology field. And what it says is that if a Mycobacterium tuberculosis enter inside an immune cell, autophagy will kill it. Okay. So this was among the first papers published to show the importance of autophagy in infection. This was from Voyo-Dertic group in US, and same time the Japanese group from Tomotsu Yoshimori group also published similar data. So what you see is one cell, and there are hundreds of lysosomes in the cell stained in red, and this is a Mycobacterium in green, maybe a cluster of them. Okay, now this is a fed condition, you are eating food. When you are fasting, all these lysosomes fuse with the bacteria. Okay, so this is what you need to imagine. There is a lot of bacteria inside the phagosome, and there is lysosome. You saw it in red color. These red color are lysosomes. The green is bacteria. But if you are fasting, all this lysosome will fuse into bacteria, which means all this phagosome and lysosome will unite, and it is going to become a lytic compartment where the bacteria will be burned, killed, and thrown out. Same fate happened for virus also. And they also studied autophagy here. As I said before, they took the DNA of the fluorescing coral reefs, and they fuse that protein which is fluorescing with autophagosome membrane. So you can see the autophagosome membrane, autophagosome inside a macrophage. So these are macrophage immune cells. Here you are in a fed state, so there is no autophagy. This is not forming any organelle. The protein is just running around. And there is bacteria, nothing is happening. When you are fasting, all the autophagosomes are covering the bacteria. So you can see that the bacteria is engulfed by autophagosome. That's why the green is localizing with the bacteria. And let's see the survival rate. In a normal fed case where you are eating high caloric diet, 100% bacteria survive. This is cell culture, okay? And when you are fasting, so much decrease in survival of bacteria. So bacteria die when you are fasting. And same thing happened to virus as well. Now there are alternative mechanisms that bacteria and virus can escape autophagy, but that's a different scenario. But let me warn you that I've read papers which says that when there is a peritoneal cavity infection, if you are fasting, the mouse die. I don't know why, but it is a scientific report. So we need to consider this, and you might know that prolonged fasting will make your intestine become like paper. Then suddenly the patients somehow, without our knowledge, if they eat heavy food, the intestine is going to break because it has lost all the muscles. It has become very thin like a paper, and if you eat food, it will break. So prolonged fasting should be very carefully dealt with. You might need to slowly increase the liquid diet and slowly solid diet. It will take a long period to pick up the intestinal muscles and intestinal walls. Otherwise there is a danger of fasting that the intestine can break. It can cause infection in the peritoneal cavity, then the patient is dead. It's very dangerous situation. But that's not all about. If there is a peritoneal cavity bacterial infection, and if you're fasting, I've read scientific papers that it causes death. Fasting causes death, but I don't know what is the reason behind it, and I haven't read more details of that. Anyway, so just a minute. So, yeah.
As you explain this, can you just correlate whether this explanation has anything to do with autophagy and COVID-19?
I cannot talk about COVID-19 now because I am explaining only the scientific published data.
Okay.
I cannot assume and say something, but I will be talking about influenza virus infection and cytokine storm, which causes fatality and death in mouse. Okay, but unless there is a solid evidence of COVID, I won't speak it. I will speak only based on experimental evidence. There is no assumption here. Okay, so let us imagine to cause a fatality, the virus need to transmit through so many difficult barriers. The first line of defense is the skin, which secretes antimicrobial peptides, antiviral peptides. There is cilia inside the lungs which will throw out the virus. And even if it succeed to escape from here, there is macrophage in the lungs which will eat up the virus and destroy them. But still, if it escape, there is humoral immunity, which is full of very toxic proteins which has the capability to destroy the virus. Okay, still it escapes. There is adaptive immune response. So this is like the army, navy, and the air force and the submarines. You have a full-fledged defense mechanism. And even if it enter inside the cell, again there is autophagy and phagocytes trying to fight it. And there is hexosamine biosynthesis pathway again trying to regulate it. So many things are there. So let us try to understand this in detail. So if it escapes from the second line of defense, there is antibody waiting to kill the virus. There is a cytotoxic immune response waiting. This cytotoxic response is like you understand that the virus infection has started spreading. So these cells will come and randomly destroy a huge area. May it be infected or not, the immune system doesn't care. It will just kill the whole area. So many. So the question is, then why we die due to virus infection? One possibility is virus escape these strategies. They have very intelligent ways to overcome the immune defense. Second possibility is that our immune system is weak because of our own lifestyle. And the third possibility could be that we have designed the wrong treatment strategy, which will suppress all the 7 lines of defense, including autophagy and phagosome and all these defense. We are doing something wrong to suppress all these line of defense. So let us try to understand this in detail. Only then the virus can succeed to give a enter throughout into finally to hijack the cell. Otherwise, it is almost impossible to cross this much barriers. But anyway, autophagy, what can stimulate autophagy? One is fasting, then growth factor, which is like insulin is less, it will increase autophagy. Then there is cytokines, which will increase autophagy. And if there is a virus or bacterial infection, this is going to increase autophagy. What will suppress autophagy? There is a high caloric diet will suppress autophagy because mTOR will be hyperactive, mTOR doesn't have a brain. What mTOR think is, oh, there is more glucose, more amino acids. Okay, it's a growing time. Let us reproduce. So mTOR will ask the cells to advance. It won't care about virus infection. So it is same as like you are living in a flat and there is a militant attack. So some robbers and militants have entered your flat, but you are doing your regular routine. You are going to cook food and eat, and you're going for job. What will happen? The militants will hijack your flat. It will take away everything you have in the flat, and it will kill you. Same thing will happen. So if there is a militant attack, you should not be cooking, you should not be going for a job, you should stop everything and prepare for the defense. And that's what exactly cell asks you to do and brain asks you to do. When you have a viral infection, brain will tell you that it will withdraw taste from the taste receptors of your mouth. You won't feel any taste when you eat food. You won't feel hungry. So brain is trying to tell you that you shouldn't be taking nutrients, you should better focus on the viral defense. So even if the bacteria succeed to cross all the barriers, it will be eaten up by the macrophages inside the phagosome. It will be burned and destroyed. Those bacteria and viruses which escape the phagosome will be shot and killed again by the autophagy machinery. So autophagy has a versatile role. It plays a role in cancer prevention, it plays a role in improving immune system, it plays a role in cardiomyopathy, aging, infection, diabetes, fatty liver, neurodegeneration. So everything comes under one thing, that if we can be in a normal healthy diet. So I will conclude part 1 of autophagy, that is mTOR is a master enzyme which is hyperactive when there is a high nutrient. It will think of growth and expansion. mTOR is inhibited when we are fasting, and mTOR inhibition upregulates autophagy, which is a detoxification mechanism. Autophagy will remove all the toxic machines accumulated, like damaged mitochondria, which is causing oxidative stress, damaged components inside the cell. Then inhibition of autophagy will cause tumors and neurodegeneration, and autophagy and phagocytosis will kill virus and bacteria during fasting. And now in few minutes we'll have a tea break, but before that I will introduce part 2, how changing glucose metabolism changes the cytokine storm. So if you remember cytokines, cytokines are hormone like proteins secreted by the immune cells. Cytokines are the language of immune cells. Immune system, when it secretes cytokines, the other immune system in the blood will understand that there is an infection going on in the lungs, blood vessel will be dilated, millions of immune cells will rush to the spot of infection, it will immediately stretch into action. A cytokine will ask immune cells to secrete antibodies, and there is a air force defense, there is a navy defense, and military soldiers defense from the land, all coming together. And when you find that there is a virus infection, if you can reduce glucose, you can control cytokine storm, because glucose is a fundamental molecule required to make cytokine. Amino acid is a fundamental molecule required to make cytokine. So in a normal pathway, glucose go through glycolysis to enter pyruvate, and pyruvate enter mitochondria to make 36 ATP. But during infection, you know that from pyruvate it is directed to lactic acid, causing an acidic atmosphere here, because mitochondria is not functioning here during an infection. But when there is a tenfold increase in glycolysis, because mitochondria cannot make 36 ATP, cell demands 36 ATP, so there's a tenfold increase in glucose uptake. From this tenfold increase, 5% will redivert for hexosamine biosynthesis pathway, and that will make cytokines. So logically, if you reduce glucose, you can reduce cytokine, and brain asks you to do that, because when you are catching cold, a virus infection, you are losing hunger, you are losing taste. This is not a coincidence. This is the brain's command to you and to your mind that you should be stopping high caloric diet so that it can function normally, because neurons are not connected to immune system. Immune system is running around, neurons are stable. So for neurons to communicate with immune system, it needs to communicate with you, and you need to stop food and you need to communicate with immune system. By stopping glucose, you might be able to reduce cytokine. So that's your inner call. So it's snacks time. I hope
Yes, sir.
Dr. Praveen, you have arranged online snacks, and I will also take questions.
Sir, we have two
Yes.
questions relevant to your first part. How
Okay.
many hours does it take to activate? I mean, how many hours of fasting does it take to activate autophagy?
So Dr. Rao has already explained about this, that from evening, let's say before sunset, if you take food, okay, then you are not taking any food throughout the night. So you have stores in your liver. Liver has storage of food. This needs to be finished before the autophagy. But of course, if there is a reduction in insulin and amino acid, autophagy will be upregulated. In mice, essentially we do 2 days fasting, and you can see a very high autophagy, very efficient autophagy with 2 days fasting. And this is usually seen in religious fasting as well. And after 2 days of fasting, ketone bodies will take over, so autophagy will come down. So 2 days is the best time for fasting, but you can have a decent level of autophagy if you take food in before sunset. Then till morning you are not taking any food, and if you are taking food for lunch, so you are getting like more than 16 hours time, then you will have a very nice cleanup procedure. And there is another interesting thing happening in your brain. There is something called glymphatic system. When you sleep, it will wash off your brain as well. So sleep is also important. If you are not sleeping, don't expect anything to happen. But anyway, these are the two things. Yeah.
Okay. And does the ketone metabolism inhibit mTOR?
There are several data coming on, and I will detail it in the next talk. It's more intense than what I'm going to tell. So I need to explain the whole pathway.
Okay, sir.
Okay. Yeah. But ketone bodies are going to play another roles. It can stimulate viral defense. There are many other things coming up. So it was published from US University very recently. So I'll detail it in my next talk.
Okay. The next question is, how long does it take for a cell to initiate the production of autophagy protein during fasting?
Okay, autophagy protein, which means recycled product. That's what you meant.
Yes.
So autophagy itself takes only 20 minutes. So if you are fasting in a cell culture, okay, when there is no amino acid or insulin, within 10 minutes, mTOR will be inactive. Then the autophagy enzyme will escape from mTOR. It will start making autophagosome. It takes 20 minutes for an autophagosome to clean up and reach a lysosome. There are hundreds of lysosomes there. Hundreds of autophagosome will be formed, and 20 minutes to clean up. And it will take 4 hours to 5 hours for the whatever is cleaned up, it should be recycled. No? So the recycling amino acids and glucose to return back to the cell will take 4 hours time. So this, yeah.
And what kind of fasting activates autophagy? It be a dry fast or any kind of fast?
I think if there is low glucose and low insulin and low amino acid, these things are going to activate fasting.
Autophagy.
Yeah.
All right.
Sorry. Autophagy. Yeah.
Yes. Thank you, sir. It was indeed a very simplified presentation with a beautiful analogy. I must really say you are indeed a storyteller researcher. Thank you for a wonderful presentation on detoxification process and the role of autophagy in our health. Right.
So it's shared. Okay.
Kindly turn to the PowerPoint mode.
Okay. Can you see that? No?
Not yet. Yeah, yes.
നമ്പറിലൊരു മെസ്സേജ് അയക്കൂ.
Fine.
I request all our viewers to kindly mute their mics. I request all our viewers, attendees, kindly mute your mics.
Okay, thanks a lot. Now, am I audible and is it clear? The
Yes, sir.
slides?
You're audible, sir. Maybe if you could increase your volume a little bit.
Yeah. Yes.
Better.
Okay.
Yes,
Is
sir.
that fine now?
Yes, sir.
Yeah. So what I'm trying to speak about is about the cytokine storm. How glucose metabolism is involved in cytokine storm, and how by controlling glucose and amino acids we can control the way immune system is responding. So before that, I will go to some basics of immunology and inflammation. So what you see here is a virus, and this is a macrophage which belong to immune system. So this macrophage cell will eat the virus. It will be chopped up in the lysosome, and a portion of this will be presented on MHC receptors outside the membrane of the macrophage, so that anybody around, other immune system can understand, okay, this is the virus. Now we need to fight this virus. It is just like you have a let's say alien attack in your town, but no one knows how an alien look like. Then you will kill the alien and put its head in front of your house, so anybody who sees that can understand, okay, this is how the alien look like. It can destroy us, so we should destroy it. So this is the same thing happening here. The virus will be presented on the receptor of the macrophage, and the immune cells called T cells will understand, okay, the virus look like this. Then it will ask the other immune cells to make antibody exactly possible to neutralize this virus, and it will also trigger another group of immune cells to make a attack on the infected lung epithelial cells. So any cell which is infected with the virus, it will be presenting the viral particles, and these cells will go and make pores there. So this is what you see here. Suppose this is a infected lung epithelial cell, it will be presenting the virus and make the immune system understand that, okay, I am infected, now I need to be destroyed, otherwise virus will use me as a house to divide more, proliferate more, and millions of viruses are going to come out from this if it is let it happen. So the cytotoxic lymphocytes will come and bind there, recognize that this is infected. It will secrete very toxic enzymes. It will make pores in the infected cells and it will kill the cell. Once the cell is dead, virus is inside, macrophage will come and eat up these dead cells. So this is a good thing in a normal condition. So virus is infecting the lung epithelial cells. So virus will be chopped up, killed, and presented. But still the virus will survive here. It will start reproducing, and the immune system will recognize that there is a virus infection by checking this receptor, and this will trigger activation of the immune system, and it will ask adaptive immune response cells to make antibodies which can neutralize the virus. And it will also trigger cytotoxic lymphocyte response, which will specifically bind to the infected cells and kill the infected cells, as I just showed here. So what antibody does is antibody will bind to the virus, and once the antibody bind to the virus cannot act. It is finished. Now this is neutralized virus will be eaten up by the macrophages and it will be killed inside. Now, what is the inflammation? So this is the defense you are seeing. Inflammation is a healthy thing for us to defend against the viral infection. It's not always bad unless it goes uncontrolled. So when there is a virus titer further increasing, the immune system realized that, okay, this is not enough. You need to call up the army, navy, and air force, and the nuclear weapons needs to be ready and prepared. So once the virus enter, this will trigger NF-kappa B pathway, which is a fundamental pathway responsible for inflammatory response. So this enzyme will go to the DNA and make cytokines. I already explained what cytokines are. You might be familiar with interleukin 6, which is causing so much of fatality due to cytokine storm during viral infection in the lungs. So it will be making cytokines. Now, just an addition that autophagy helps to control this cytokine release by controlling this driving wheel like thing called inflammasome. So autophagy can actually act here to fine tune cytokine release. But anyway, I'm not talking about autophagy here. Let's talk about cytokine. So once cytokine is released, what it does is it's going to dilate the blood vessels. It is going to communicate with millions of other immune cells nearby. It will recruit millions of immune cells to the site of infection in the lungs. Then it just goes crazy. It will keep secreting more cytokines, and then it will amplify the antibody response, but it will take at least 1 week to come. It is just like Air Force. The first the land army goes and fight, and only as a second line of defense the Air Force come. So antibody is like that. It comes after 1 week. But the initial fight needs to be handled by the macrophages and the innate immune cells. So interleukin 6, the cytokine, it will increase vascular permeability so that from the blood, millions of immune cells can come to the site of infection. It will increase antibody immune cell recruitment. Then if goes abnormally, it can cause fibrosis and fluid accumulation in your lungs. Then you cannot take oxygen in the cells. Then once virus titer increase further, goes out of hand, immune system gets crazy, it will come and start destroying the whole area of the lungs. So you can find that one whole area of the lungs is dead, and fibrosis will start, and there will be lack of oxygen, and it causes fatality. So let us try to understand what it is all about. I said that in a normal case there is some virus, but that can be easily handled by the immune system. But when there is more virus, because you are taking a high caloric diet during a cold or viral infection, so immune system cannot really handle it in a fine-tuned manner. Virus autophagy won't be there, so phagolysosome fusion will be very weak, and virus will be increasing. And this is an air sac of the lung. This is blood vessel. So virus titer is already increased, and there is macrophage trying to reduce the cell, but it's not happening. So more immune cells will come there, and it will cause a traffic block, and it will start secreting very toxic enzymes. And as you remember, it can destroy the cells. So these cells can destroy the infected cells. So it will start randomly killing everything. It is like you realize that a town, every 3 house has a militant which can destroy. So it's going to spread everywhere. Now there is a moment that we will think that every house need to be fought individually without by saving the other house. But immune system doesn't think like that. Immune system will decide to kill the entire area by just one bomb or something like that. So it is going to destroy the whole alveoli sac, so that let us finish off. This is enough. And this will cause a hole. And then lot of fluid will rush into the air sacs, and fluid will start accumulating because of excessive cytokine storm. And there is a huge cluster of immune cells coming to recruit and trying to rescue, but not happening. So this is what happens in a mild infection where there is few virus and you are in a low caloric diet. Autophagy is successful to kill the virus, and phagolysosome fusion is successful to kill the virus. Macrophage can easily finish off the virus infection. But when you are in a very different lifestyle, you cannot kill the virus, and then there is a havoc. You have a lot of immune cells coming here, and in the air sac of the lung, fluid starts accumulating, the membrane is destroyed, and it is a catastrophe. And then you can see that lungs is filled with fluid, and they cannot breathe, and you end up in fatality. Now, the interesting part is that if you control glucose and amino acid, you can actually control cytokine storm from the upcoming evidence from the published papers. But before going to that, I will briefly cover basics of metabolism. So this is carbohydrate. So am I audible? There is clarity in my talk?
Yes, sir, you're clearly audible.
Okay, so we have carbohydrates, triglyceride fats, and proteins. This will be broken down into glucose, fatty acids, and amino acids, which is reaching in the blood, and this will enter the cell. And what all these have in common is carbon chains. If you remember my early presentation, it's all made of carbon chains, and cell use this carbon chains. When these carbon chains are broken down, it can make energy, because plants use sunlight to join the carbon chain. So there is an investment of energy using sunlight, and that energy is captured between the carbon chains. And so that is an investment of energy. Energy cannot be made, it can be transferred from one form to another. So that chemical bond energy, when it is broken down in the mitochondria, it can be made to ATP, or chemical energy molecule. So glucose, when it enters the cell, so this is the cell membrane. When it enters cell, it passes through glycolysis, and the glucose will be broken down into two, which will be pyruvate. Pyruvate will enter the mitochondria. It will pass through the TCA cycle, where finally it enters the electron transport chain to make 36 ATP from one glucose. But when there is a virus infection or cancer, this cannot happen. Pyruvate is redirected to lactic acid. So this happens in a very small range. Majority will be this pathway. Lactic acid is formed, there is a high acidification intracellular and extracellular space. Acidity further activates cytokines, and there's a whole mess. And you get only 2 ATP. Now you need to upregulate the glucose tenfold to match with the 36 ATP requirement. So this is a bit advanced about glycolysis pathway. Glucose enters the cell. Glucose is converted to pyruvate, which is actually breaking down of carbon chain. It enters the mitochondria and goes through the Krebs cycle, and finally enters the electron transport chain to make ATP. During infection, this step is blocked. Pyruvate cannot enter the mitochondria, and that is Warburg shift. So in a normal case, glucose enters the cell membrane, and inside the cell it's broken down into pyruvate. Pyruvate enter mitochondria, where 36 ATP is generated. In cancer cell and during virus infection, this cannot happen. There is only 2 ATP made, and there is an acidity, and only 2 ATP made to match the requirement. There is tenfold increase in glucose. Out of this tenfold increase in glucose, 5 to 10% will be rediverted to hexosamine biosynthesis pathway. And this is very important. This pathway is responsible for making cytokines. So let us try to understand in detail. In immune cells, uninfected cells, glucose forms 36 ATP through mitochondria. Infected macrophages, glucose form pyruvate. It cannot enter mitochondria, it forms lactic acid. And then there is an increase in glucose uptake tenfold. And before forming lactic acid, this is rediverted. So virus itself can rewire the metabolic pathway, causing pyruvate to lactic acid conversion. And so there is a virus dependent metabolic reprogramming, and in addition to that, you are taking high caloric diet during a virus infection. So immune system almost give up at this condition. So this is what I was telling. Virus infection causes metabolic reprogramming towards Warburg shift. There is tenfold increase in glucose uptake, and from this glucose increase, 5% will be rediverted to hexosamine biosynthesis pathway, which makes cytokines, right? So when you catch cold, you might experience loss of taste and hunger. This is that brain is demanding you to stick to a low calorie diet so that it can communicate with the immune system through you, through the metabolic molecules. It communicates with the immune system to decide how much cytokine need to secrete it according to the virus abundance. So if you ignore this brain signal and your inner core, you are going to end up in cytokine storm and lung fibrosis. So this started with the initial finding that in diabetic patients in 2000, 1999, 2000, Cora group understood that diabetic patients who has hyperglycemia, they also have high cytokine storm. Right? TGF beta causes lung fibrosis. So they try to understand why, and they found out hexosamine biosynthesis pathways behind this. That when there is more glucose, instead of going into 36 ATP, it is reconverted to lactic acid, and only 2 ATP is made, because of which there is tenfold increase in glucose uptake. And from this tenfold increase glucose uptake, 5% is rediverted to hexosamine biosynthesis pathway, which makes amino sugars, which means amino acid plus sugars combined. This is a very fundamental molecule required to make cytokine secretion. So these are the enzymes in this pathway of hexosamine biosynthesis pathway, and a very important enzyme is OGT, which brings the cytokine to a modified form using the amino sugars, and only this can be secreted out in a functional form. And one more pathway was discovered, that glucose, when it is rediverted to hexosamine pathway, it also enters protein kinase pathway, and then it will go to nucleus and increase TGF beta cytokine secretion. So this brings us to a very important paper published very recently in Science Journal, that when there is influenza infection and you have a high caloric diet, it will cause cytokine storm based on the increase in expression of cytokines. Okay, so based on this, they found some experiments. Let us go through that. This is a normal condition where you have very less TGF beta or cytokine. This is a Western blot where you can find proteins. You can detect proteins. So you see detect very little protein. So this protein band will be formed of like thousands of proteins coming together in the same line. So when you have an increase in hexosamine biosynthesis pathway, which means this rediversion, there is increase in cytokines. And they also found that when there is an increase in glucosamine, which you might remember, glucose plus amino acid makes glucosamine through hexosamine biosynthesis pathway, there is an increase in the enzyme activity and increase in cytokines. So increased glucose uptake and utilization in immune cells serves as a hallmark feature of acute inflammation. Let us try to read this, a brief, bit complicated, but give me a minute, please. So let us try to understand this. What they are trying to say is, yeah.
Yeah, we have 20 minutes remaining for the session.
Okay. No, I just want, now I'm free now. Is it okay? I continue?
Yes, sir. 20 minutes more.
Yeah. So let us try to read this. In this study, we demonstrate an essential function of hexosamine biosynthesis path, which is the pathway leading to cytokine secretion. Okay, associated with glucosamines, that is glucose with amino acid, which you are eating. Signaling influenza virus induced cytokine storm. So when there is a virus infection and you have high glucose and amino acid in your blood, you are going to end up in cytokine storm, according to this article published in Science Advance. Now we determine that the mechanism behind this induced by OGT, which is the enzyme in the hexosamine biosynthesis pathway, which is upregulated with an increase in glucose. So analysis of clinical samples revealed that, clinical samples mean human patients, revealed that the N-acetyl glucosamine, higher levels of pro-inflammatory cytokines are correlated with high blood glucose in influenza virus infected patients, which means when there is high cytokines, they also found that those patients have high glucose in the blood. And the patients which did not suffer from cytokine storm, they do not have high glucose in the blood. So there is a direct correlation between glucose concentration and cytokines, which is causing death during viral infection. So we identified a molecular mechanism by which hexosamine mediated pathway of glucosamines function in influenza A virus and highlight the importance of glucose metabolism in influenza A virus induced cytokine storm. So this is their model. When virus infects the cell, it is going to rewire the glycolysis pathway and it will enhance the hexosamine biosynthesis pathway, which will upregulate OGT enzyme, and through mitochondria, it is going to advance another pathway of I kappa B, and it will go to nucleus to increase pro-inflammatory cytokines, which is going to secrete cytokines heavily. And now you know what cytokine does. If cytokines are secreted abnormally high, brain cannot stop it, because brain already told you by stopping your hunger that you're not supposed to take, but you ignore it. Then brain has no control on immune system. Immune system has to manage by itself, but immune system understands only metabolic molecules like glucose and amino acid and virus. It cannot understand brain. So immune system and brain language was your mind, and you have broken it. Now the cytokine storm is going to peak, and this cytokine storm is going to heavily recruit billions of cells, immune cells, to the spot of infection in the lung, and this will also expand the blood vessels. Lot of fluid is going to accumulate in the lungs, alveoli, air sacs, and the patient ends up in lung fibrosis because oxygen is not coming, the cells are dying, and the immune system thinks the virus is expanding without, because autophagy is inactive, phagocytosis is inactive, and everything seems giving up. And this excess cytokine storm also inhibit antibody production. So you realize that the Air Force is not going to come as a backup. Like antibodies are not going to come in 1 week. So immune system gets its best full-fledged defense. So let's see the clinical data. But these are done in mouse. What they've done is the orange bars. So x-axis shows different cytokines, interleukin 6, interleukin 8, tumor necrosis factor, and y-axis is the amount, picogram per ml. So influenza A virus plus glucosamine, which is the glucose plus amine, amino acid. If you give high glucosamine plus influenza infection, you can see that they have high cytokine storm, yellow bar. All the cytokines are high, but only influenza infection. But the mouse is taking diet. but a low, not a fasting, but normal diet, then you don't see that much. You see the red bars. So when you have a high glucose and amino acid, there is a threefold increase in cytokine storm, and this is far than enough to kill an animal. So let's see the survival rate of this animal. So in a normal control, like only glucose without infection, 100% animals survive. Okay, but when there is a influenza virus infection, after 15 days, that is 2 weeks, 40% of the animal die. But if you have high glucose and amino acid plus an influenza infection, 100% animal die in 15 days. That is mouse. In human being, only 4 to 8% people die. Remaining 94% survive, because human body is made in a much advanced level that even if how much you torture your immune system, still the body has some kind of backup mechanism to make you keep alive. And this is the reason. Otherwise, you will end up 100% fatality rate. So what they found is the patients infected with the influenza virus has high glucose. Okay, and this correlates, high glucose correlate with interleukin secretion, and they check in a Western blot that all the enzymes regulating the hexosamine pathway are high. So there is a direct correlation between glucose levels and inflammatory cytokine expression in human patients. Okay, now this is again a Western blot. All these side is healthy individuals. This is the influenza infected patients. You can see that enzymes are high in infected patients with high caloric diet, but you don't see those enzymes active when there is no infection. Okay, now their aim is to make a drug, not fasting. They are not your group. Their aim is to make a medicine which can prevent it. So they inhibit OGT, that is the enzyme here. So if you inhibit OGT enzyme, hexosamine biosynthesis pathway cannot complete. So cytokine secretion will be tremendously reduced. So that's what they did. The orange line is OGT inhibited animal with virus infection. You see that 100% mouse survive even though they are virus infected. But the control infected mice, you see again 30% animal only survive. 70% die in 2 weeks. Okay, so but this is for a drug, but if you actually reduce glucose, you can actually inhibit hexosamine pathway, because glucose is the initial product to activate hexosamine pathway. Now interestingly, they observed that virus titer is less when hexosamine pathway is inhibited. Why is that? I do not know. Maybe when there is low glucose and amino acid, autophagy is activated, phagolysosome fusion is high, and there are many other pathways which we do not know. Or cytokine storm is causing some. Anyway, virus titer is less when you don't have hexosamine biosynthesis pathway. There is low glucose and amino acid. So reduced virus titer and reduced cytokine level when hexosamine biosynthesis pathway final step is inhibited. So this is what it's all about. So once you have high caloric diet, high TGF beta cytokines, it will cause cell death, apoptosis. It will cause lung fibrosis. It will inhibit immune cells to make antibody. So when there is a high cytokine storm, the second line of defense, the Air Force, is not going to come for backup. So there will be heavy fatality from the immune system side, and the lungs and the immune system both will be dying. That's why TGF beta is regarded as the titan of lung fibrosis. And I already explained what happens in a hard case when there is cytokine storm. You are in a high caloric diet, hexosamine biosynthesis pathway is high, there is high cytokine storm, millions of immune cells are rushing to the site of infection of air sac, and this will also cause fluid accumulation, and cells will start dying, and there is fibrosis. So this is the initiation of fibrosis, and now the lung is dead. And another interesting thing is finding that temperature or fever can increase NF-kappa B pathway. This is an inflammatory response, very important to fight the virus. So inflammatory response is not bad. It is very important to fight the infection. So if inflammatory response is high, you have a fever, you will have a good NF-kappa B pathway, and then the immune system can efficiently fight the infection. So even if all these line of defense are there, still people die due to virus infection. As I said, virus may have strategies to overcome immune defense, or we are intentionally making a wrong treatment strategy which will suppress all the lines of defense. So let us try to understand what are the lines of defense. The first line of defense is obviously the lung epithelial cells, the mucosal lining. So let's say if there is a wound in the lung mucosal lining, how can a wound happen? Suppose you are in a split AC room. The moisture content will be 70 to 80% inside the room, but overnight air condition. Next day, the moisture all night, you will have a room humidity of 35 to 40%. So this will make your whole mucosal system dry, but not everybody. Some type of people. So I cannot explain this with a normal logic. According to Ayurvedic principles, some people have a sensitivity to dryness. So those people, their lung, other people will be okay, but those people, their lung will get dry, mucosal system membrane will rupture. This gives a easy chance for virus to enter. Then you are in a closed room, even if you keep like 10 meter or 20 meter distance, you are in a closed office, there is a virus. It is going to circulate all over the room, even if you keep distance. So it's going to enter many people. But if it is an open window room, the virus will not spread that much. Anyway, that's not our topic. And if there is a diesel and dust mix, you might realize that like in Japan, during Sakura season, huge population of people catch cold, they started coughing. Then initially they thought it's a virus infection, but they realized that people who live near the national highway are more prone to catch this catching cold and all. Then they realized that during Sakura season, the pollen grain mixed with the diesel in the national highway. So mix with diesel and dust, and this forms haptens, which the immune system consider as a emergency signal. So it looks like virus for the immune system, and there is a very high inflammatory reaction, but it won't be a virus infection. But anyway, so environmental factors do contribute, but that is just initial thing. But real story starts inside. Once the virus enter inside the cell, the brain will tell you to stop eating. That is, you will be under anorexia, and you will lose your taste, and you will feel feverish. Feverish is you didn't get the temperature, but you already feel feverish, which means it's going to come. Infection has started. Brain is telling you that, but since you cannot taste, you will start eating tasty food like ice creams or high caloric diet or any carbohydrate or high fat protein diet. What's going to happen is autophagy is going to get inhibited. Then phagosome lysosome fusion is going to inhibit. Then when there is temperature, fever is coming, immediately you will be given a tablet to suppress your fever, and you will be never be allowed to increase the fever. So fever is so important to increase NF-kappa B pathway in the initial stages of viral infection. So when the virus is entering the cell, when there is a fever, NF-kappa B pathway is active, immune system can really wash off the virus. But you are suppressing the fever, thinking that the brain will be damaged. So we need to really see a study at what temperature, what percentage of people, animals have brain damage. Then we can conclude that, okay, after 41 degree, maybe the brain damage happens, so we can control that temperature not going above. But totally suppressing fever will definitely inhibit NF-kappa B pathway, and this is going to create trouble. So virus succeeded to enter the cell. Okay, then because it finishes the initial lines of defense, and now you give chloroquine. What chloroquine does is very simple. Chloroquine will inhibit the function of lysosomal enzymes. So virus is inside the cell, and as step 1 virus is phagocytosed, and step 2 virus is killed inside the lysosome. But chloroquine will inhibit virus killing. No data, but we know that chloroquine will inhibit lysosomal activity. It will also inhibit one major thing, the viral antigen presentation by MHC. This antigen presentation is very crucial for the immune system to understand that there is a virus infection. Then only this will cause a heavy recruitment of immune system here and fight the virus. But this is not going to happen. This whole process is suppressed. So virus successfully cross this line of defense because you are providing chloroquine. But again, we don't know the benefits of chloroquine. This should be clinically analyzed and compared with the placebo trial. That is, you have virus infected animals who are not taking chloroquine, and you have virus infected animals with taking chloroquine, and what percentage of animals survive. You know that 93% of people survive the viral infection. Only 3% die who are about to die, or people who are already dead, you take a sample, you will find them positive. So who are dying, they have the positive for the virus. So 97% survive. Out of that 97% survive, out of the 3% who died, how many percent can chloroquine rescue? This need to be statistically significantly analyzed. So virus has passed the initial line of defense, which is the killing of virus and presenting it on the macrophage so that the immune system can make antibody against it. But now immune system cannot make antibody because you have inhibited antigen presentation, and B cells cannot make antibodies. So the second thing you are going to give is chain terminators, which are some of the popular antiretroviral medicines. What they do is they are going to bind to any dividing DNA or RNA. So they are nucleotide analogs. They will bind to viral RNA, which are dividing. So this is going to effectively reduce virus titer. But mitochondria also has a DNA. This we need to remember. Mitochondria is dividing. Hundreds of mitochondria in the cell very effectively functioning during a virus infection. Mitochondria is the most important machinery during a virus infection. It need to advance its process, and it need to divide to make new DNA. And all the mitochondria, after heavy hard work, they need to be recycled. So the chain terminator will bind to viral DNA. It will inhibit mitochondrial function. This is same as you are in shutting down the electric power in a town when you have an enemy attack. So if you don't have electricity in the town, how will you fight an enemy? This is the same situation happening in a macrophage or immune cells when you take chloroquine plus chain terminators. So definitely the immune system will be suppressed. Maybe cytokine storm will come down because immune system is totally suppressed. So 3% only die, 97% people survive. But we really need to understand this. If we are suppressing fever, we are going to suppress NF-kappa B pathway, and we are going to mess up with this huge, complicated, extensively advanced enzymatic pathway, which is going to bring very effective immune defense against the virus. We are going to suppress all these. Still, 94% people survive. Why? Because God made human body in such an advanced way that 1,000 times better than a spaceship. When 100% mice die, we survive. So I will conclude part 2, high glucose and amino acids. So high caloric diet during a viral infection increases viral titer. Okay, we don't know it is because of autophagy inhibition, but it increases viral titer. High glucose increases cytokine storm and fluid accumulation, lung fibrosis, and 60% fatality in mouse because of increase in interleukin 6 and TGF beta. Fever suppression inhibits NF-kappa B pathway, and this allows virus to further advance and pass through all the effective barriers of the immune system. High caloric diet suppress autophagy and phagolysosome fusion and inhibit NF-kappa B lysomal MHC presentation. So chloroquine shuts down the viral presentation, so immune system has no idea how much virus is there. They won't be making antibody, and you are shutting down the powerhouse of the cell by taking antiretroviral medicines. But I would not recommend following this in your patients. These are just mouse experiments, and this is just our philosophy. Unless there is a proper clinical evidence, you should not try it on patients. So only after human trials, you are allowed to try them in patients. So now this is my part 2. Third part, I will explain how the ancient medical herbal medicine practitioners and sages in the Himalayas and South India understood which medicine, which herb should be used to treat one particular disease. So I'll come to it in my part 3. I'll take a small break and open to questions.
Yeah. So thank you for summarizing. As in naturopathy medicine, we rightly say, why is it important to respond to body's signal of loss of appetite and demand for rest during any illness, and how acute inflammation is body's way of cleansing itself. Thank you for stressing the importance on introducing fasting or calorie restrictions during the viral infections or to curb the inflammatory processes, especially to reduce fatalities by preventing the cytokine storms. So we have a few questions here.
Yeah.
It says, as most of the immune boosting foods we know are high calorie diet, what is your take on these immune boosting foods during a viral or bacterial infection?
So can you explain what is that food? Like
immune boosting foods, you mean to say now generally we talk about fat, good quality fat, or mutton soup, or any type of vegetarian soup, basically roots, carrot and all that. So what is your take on these high glucose?
So I feel that we need to listen to what our brain is trying to tell us, and the initial days are very crucial. So fasting is very important, and so this part I cannot tell. Only doctors can tell, like you, who treat patients. I work with animals, so it's very different system. So I would not answer this, because you know which is the best treatment protocol which can effectively cure a virus infection. And yeah, I would opt out of this question. But let's see what happens in the cell. When there is low glucose and low amino acid, you can reduce cytokine storm. So it means that if you're taking a high carbohydrate and high protein fat diet, you have a trouble. But soup doesn't have a high calorie. So if you look into Professor Walter Longo's work, he has put fasting mimicking diet. So this is a very low calorie diet, and this seems to have a similar effect of fasting. So I think this kind of soup, vegetable soup and all, this is very low in glucose. So this can, maybe it is effective during the infection, because you cannot go for a long-term fasting. So soups are advisable, and it should, I think, only from your experience, it can be made into a proper protocol. And now we have a huge group of doctors joining this meeting. Maybe we all should come together and discuss which is the best protocol of diet to treat a particular virus infection. So Praveen's website is there, so there should be an open forum where you can discuss the best protocols based on the case studies, human trials. I am only a molecular biologist doing cell culture, so I won't be able to answer this. Yeah.
Okay. And the next question is, what is your take on immune enhancing natural supplementations, especially vitamins and minerals?
Now, of course, vitamins and minerals are very important. So what happens is, let us say, when human beings were there in nature, we were surviving on fruits and vegetables and meat without boiling. Like, we were not taking rice and boil and eating. So that is what evolution has made us. So our enzymes are only made to digest that. But when we start modifying our protocols, like we took carbohydrate, so we took rice and boil and eat. So this is, I showed in the beginning, starch is a very complex glucose chain. So you are bringing like 5 to 10 fold more calorie into your body, and your enzymes are not evolved to balance it. So when you take more food, you are not taking enough vegetables and fruits. So you have a lack of vitamins and minerals. So what is the essential amount of vitamins and minerals you need to eat is, when you don't take any kind of boiled food, how much raw food you will eat. Every 1 hour I'll be feeling hungry, right? I'm not taking rice. I'm not eating any chicken fry or ice cream or anything. Every 1 hour I will be feeling hungry. I eat a banana, in half an hour I'll feel hungry. I eat an orange, half an hour I'll feel hungry. So we will be continuously eating fruits and vegetables, which will provide us so much of minerals and nutritions, but we are not eating that much. When we eat high calorie diet, we cannot eat that. So this is the imbalance happening. So we need to take that much food. So we need one plate full of vegetables, sprouts, nuts, all these, which we are not eating, because when we eat high caloric diet, little bit itself we are full, so we cannot eat that. So we do have a huge mineral and vitamin imbalance, which we need to balance with raw diet. And I don't know about the tablets. Okay, I haven't worked with that. Yeah, but this is just my philosophy. Yeah. Yes.
Yeah, in case of fever, as we said, we give medications to reduce fever. What about the natural, the question is, what about the natural methods like hydrotherapy to control the temperature? Does it affect in the inflammatory process?
So we need to understand that fever is decided by the brain and the immune system, and they know very clearly what to do. We don't need to teach them. They have an idea how many virus is there in your body. They know the counts. Based on that, they increase fever. Okay, everything is based on that. So we just need to obey them and lay down. And so there are reports that when there is high fever above 42 degrees, you can have brain damage. So why does that happen? Because you might be taking food, like you have a small baby, she need to drink milk, otherwise it's very difficult to manage. She will be drinking milk, and now the fever will go high. So these kind of situations happen. Then you need to control the temperature, otherwise you will have brain damage. But we need to see a proper clinical study at what temperature brain damage happen. So that is required. But the present belief is that whenever there is fever, immediately it should be suppressed, and that will cause brain damage, that's a problem. But I feel that you have a proper standardized protocol that you cool the brain by providing a wet cloth, and that works. It cured the disease successfully in few days. So you should stick to a protocol which is working. So I don't need to change that. You have a extensive experience in this. So I don't find a problem with providing cooling for the brain, because the infection is happening in the lungs, not the brain. So lung is again hot. So you are just protecting your brain. But some suppressive therapies are there, like immediately you try to suppress temperature by keeping ice or something. This is very unnatural. I don't recommend these kind of things. Yeah. Okay, sorry for the delay. Please wait. Yeah. So what I was trying to say is that, can you see my slide? It's loading. Yeah. Can you see the slide?
Yes, sir.
So immune system has 7 to 8 very strong line of defense, and the virus succeeded to get a free pass to enter the cells and kill the human body, because we have a treatment protocol which will suppress every step of this immune defense. Like what I explained is, we have suppression of temperature, which will suppress the inflammatory response, NF-kappa B pathway. Then we give some medicines which will inhibit the lysosome function. So the virus cannot be killed, it cannot be presented to the, and then antibody cannot be made. Then you are in a high caloric diet. Nobody talks about the kind of food you should eat during virus infection. Then you are also inhibiting the humoral immunity by taking the kind of diet, and then you will also suppress antibody response because of the medicines you are taking. And we are also suppressing autophagy, and hexosamine biosynthesis pathway will be abnormally increased, which will cause excess cytokine storm. So now I will come to my last part of the talk. Please give me a few seconds. So the fundamental question is, out of this millions of plants around us, how the ancient sages and Ayurvedic physicians or Aristotle understood that one particular plant can cure one particular disease. So there is a very intense ancient research behind it, but probably all of them were burned off in the ancient universities we had. So we don't have any knowledge about this, but now we need to re-engineer Ayurveda using molecular biology to understand this even to a small extent. And we don't have knowledge of the rejuvenation therapy called Kayakalpa, or this might be actually rejuvenating the stem cells. So stem cells are the mother cells of our body, which can provide more immune system cells. So like, if you're going to exhaust your stem cells by artificially proliferating the stem cells, you are providing an artificial chemical or alum or adjuvant to induce hyperproliferation of your stem cells or immune cells. I do not know, there is no evidence, but stem cells are limited. The number is limited for throughout your life. It is like you have 10 million rupee for your whole life. Now you need to use that money to complete your whole life. You don't get anything more. So stem cells are like that. It can divide. So they are mother cells. It will divide into two, and one will become another mother cell, but the other will become the immune cell, which is fighting the infection. But that mother cell, which is a battery, it is like a battery. It is going to finish when you are old. That is why in liver, old people's liver, it cannot regenerate because they run out of stem cells. The immune system is weak because they run out of stem cells. So even if you put vaccine into a 75-year-old person, you cannot expect that they will make antibody because there is no enough stem cells. And suppose you are artificially exhausting the stem cells by providing some artificial proliferation inducers, then every year you are providing that in old age. So maybe this battery finish off fast. Then you won't have any more enough stem cells to fight an infection. We might all become immunodeficient in the upcoming years. Like in 10 years, if every year you are forcefully, artificially making your stem cells to divide and proliferate in billions of cells for a infection which is about to come, you are preparing the body for that infection. But every year you are doing that. I do not know, there is no clinical evidence, but this is something we need to think of, that your stem cells may run out. And in 10 years, we will have a new disease, which will be some huge, heavy immune deficiency disease. I do not know. So, well, let us finish off with something more peaceful. Let us go to the basic elements of a disease formation. So, am I audible clearly?
Yes, sir, you're audible.
Yeah. So both Aristotle and Ayurvedic physicians, and if you look into ancient Hippocrates textbooks, it is available online. I would recommend you should read that. They say that an imbalance in 5 elements is the fundamental cause of a disease. So this is not that easy to understand for us, and it's not what we are thinking. It is very extensive in molecular level, and it's not possible to reverse engineer this in like 10 to 20 years. But I got a tip of an iceberg of this, which I wish to share with you. So please try to work more on it and share your ideas in the next meeting or in Praveen's website as a common platform. Anyway, so space, or akasha we say, plus air is considered as forms vada, or it has the characteristics of dryness and cold. So if you are not sleeping for 2 days, Ayurveda says your vada dosha increase, become abnormal, which means you have a dryness in molecular level, and you might be experiencing cold in your legs. So your vada dosha increase when you have an imbalance in Akasha and Vayu, but not so easy to understand for me. And let us try something else. Earth plus water is Kaba. So it is not really cold. It's more catching cold. It's more extensive than in terms, and I have no idea what it is. But one of the characteristics is the fundamental cause of catching cold. And for Ayurvedic physician, balancing a Kaba is nothing. And balancing a virus infection is nothing if they can be treated from the right beginning. But the unfortunate part is that these patients come at the last moment when all the other treatments become impossible. So anyway, the tricky part about this, and the most important trick, is that they extensively classified the taste of the plant, because in early days there is no molecular biology laboratory. They cannot classify what is there in a plant. But what they can classify is the smell and taste of a plant. Okay, so if you look around the nature, you can see that the bad things are black, the bad things are smelling bad, right? So it has a smell. So there is an evolutionary relationship to all of these. The good things are bright, like apple, carrot, grapes. All these are bright, attractive colors. Let's see the bad things, like a decayed food full of fungus. You will find it so dirty and smelly. It is black and brown, and so that's not for us to eat. But so nature has a language to communicate with us. This we cannot understand, but the sages understood the language of nature, and sages have a language to communicate with nature. And let us say now molecular biologists also have a language to communicate with nature through our experiments. So we do an experiment and ask a question to nature. Nature answer it through the result. So this is a way of communicating with nature. Anyway, so the interesting part here is a bitter taste of a plant can increase dryness in your body, but it can reduce kapha, or the fundamental cause of catching cold. And the opposite happens with sweet taste. If you are taking like milk plus biscuits plus some ice creams when you catch cold, you are definitely going to create a kapha imbalance and you are going to increase your cold. Let us try to understand the molecular terms behind it. A very recent paper published in clinical investigation showing that bitter taste and sweet taste receptors regulate innate immune response during an infection. So let's see what this is all about. Bacteria secretes bitter products. So immune system knows bacteria is bitter. I told you that there is a communication between nature and our inner self, and this is this kind of communications. So even though we do not know, the lung cells can identify that there is bacteria and there is this many number of bacteria, and this is the type of bacteria. It is a virus or bacteria, they know. And this bacterial products will bind to a chemosensory cell in the lung. This is binding to T2R receptor and it will promote calcium outburst from endoplasmic reticulum. Calcium is a signaling molecule which can spread throughout to many cells nearby, and this will upregulate secretion of antibacterial peptides which are going to make holes in the bacteria and destroy it there and then. And it will also secrete antiviral peptides from the immune cells, and it is going to kill the virus on the spot. Okay? But if you're taking glucose, according to Ayurvedic philosophy, sweet taste will increase kapha. Or if you're taking, what is behind the sweet taste? It is the glucose, right? So what glucose does is, glucose molecule will bind to the receptor called T1R receptor. It will inhibit calcium outburst from endoplasmic reticulum, and this will inhibit secretion of antiviral peptides. So virus will easily cross the initial barrier. So nobody is telling you that you need to restrict glucose. So virus get a free pass to enter the initial line of defense, that is the lung epithelial cells and mucosal layer. So this was tested further with some experiments. This is an LB plate nutrient-rich media where bacteria can grow. So you give some saline, phosphate buffer saline, and you give a bacterial infection. Then you take the mucosal extract after giving saline. You mix in a tube with bacteria, and bacteria will grow. At the increased dilution, you can see bacteria grow as colonies. So bacteria grow successfully. But if you give dinactonium, which is a very bitter compound, I do not say all bitter compounds will inhibit virus. Okay, again there are extensive classifications in this, which I have absolutely no idea. The Ayurvedic medicines, like you might know that it's Amardotram Kashayam, which is recommended during viral infection. And if you go through, several Ayurvedic physicians recommend bitter medicines during virus infection. So dinatonium. If you give mouse dinatonium, then you take the nasal extract, you put it in the bacterial tube and then plate the bacteria doesn't grow. Why? Bacteria die. Okay, because dinatonium is going to make lung cells secrete antibacterial peptides, which will make holes in the bacteria, it will kill the bacteria. So this is a staining to see bacteria. You can see bacteria in green. All the live bacteria stained in green. Red bacteria are stained in dead cells. Dead bacteria are stained in propydium iodide, which is red. So in saline control, you see most of the bacteria, majority of bacteria are live. But when you give bitter compound, and all the bacteria are dead. You don't see any live bacteria. So powerful, the human body's secretion. And this is a signal which glows when there is calcium outburst. So you give the bitter compound, within 8 seconds, one cell start glowing, which means there is a calcium outburst. So this will glow when there is a calcium outburst. And within 11 seconds, it spread to like 20 cells. In 19 seconds, calcium has spread to like 100 cells. And you will find calcium everywhere in the lung within minutes. And these are going to secrete antiviral and antibacterial proteins, which will destroy the virus and bacteria, shoot at sight. That is the concept of immune system. Then, so this is what you see. But if you take glucose along with it, you don't see that anymore. The graph is down. So, and if you take dinatonium, that is the dinatonium alone, you have high peak. But if you take the beta compound along with glucose, so these days, Ayurvedic doctors are afraid to ask you to go for a strict diet like patya, because once you start telling you cannot eat meat or you cannot eat rice, the patients run away. But in my childhood, I used to remember when I was a small kid, the Ayurvedic doctors were extremely strict with the diet. You cannot get any caloric diet during the first week of infection. So this is the concept behind it. Even if you take bitter medicine, if you are taking glucose, high glucose carbohydrate, you don't have any kind of signaling and you don't have any kind of bacterial destruction. And salt. If you take salt, it neutralizes the effect of this bitter compound and it is increasing, but it determines, on each case it varies. Okay, this is in the case of bacterial infection. So the colony forming unit of bacteria is 0 when you have dinatonium, bitter compound, and in buffer saline control, it is 100% bacteria are alive. But if you put salt, sodium chloride, you can see that as you increase sodium chloride, the bacteria growth increase. So sodium chloride, salt also inhibit this. So this is how they identified. Salt will inhibit a particular tridosha. Sugar will inhibit, sugar will increase kapha. Salt will increase kapha, but bitter compound will decrease kapha. So this is how they make this whole formula. And here again you can see beta defensin is antimicrobial peptide. It is highly secreted, nanogram per ml, in bitter compound treated condition. But when you mix bitter compound with glucose, the animal cannot secrete this antimicrobial or antiviral compound. But if you mix it with coffee beans, you see some rescue, but I won't recommend that. You should stick to the regular protocols, because coffee beans is, again, it will have a medicinal effect. I do not know, but it is like you have a donkey who is tired and you are beating the donkey to run. So the donkey will run, but eventually it will make him more weak. So this is just my philosophy. You don't need to apply it in your treatment. So Coffee, something like that. If you are really weak, you need rest. Body is asking you to take rest. You drink a coffee, you are sleepy. You have low oxygen in your brain. But you take coffee, suddenly you become active. So it is like a stimulant. So you have a donkey who is tired because of heavy load work, and now it needs some sleep. But you beat him, it will start running and working again. It is advantageous for you for the temporary period of time, but eventually it will make you more weak. It will make the animal more weak. So that is just my philosophy. But anyway, you understood the concept behind it. So defensin is an antibacterial and antimicrobial compound which is secreted during bitter hormone. That is the conclusion of this. So that is about Kaba and what happens in Pitta abnormality. This is another most interesting part. So Pitta is characteristics of fire and water. So what are the properties of fire and water? So heat, fire has redness. What is it? So inflammatory response, you will see redness, you will see heat, you will also see inflammation of water accumulation. That's why you see an enhancement of your tissue. So maybe, I am not sure. Pitta abnormality means Agni plus water. It could be inflammatory response. Then how to reduce this? You have a huge classification of plants with some kind of taste like ginger taste or a special type of taste which can inhibit or control this kind of cytokine storm or control lymphocyte infiltration, which is ginger, compounds of ginger. So just normal ginger may not work, but if you have the dried ginger after boiling, that is more effective in clinical studies. It can reduce lymphocyte infiltration into the lungs and liver. So there is a long list of things: ginger, black pepper, cloves, turmeric, tulasi, tumba, panikurka, mints, a huge combination of very strong herbal medicine, which is going to balance pitta. Well, I am not an expert of this, but what can we do to reverse engineer this is to understand the molecular basis of this. What are the compounds? And so you know that bitter taste, excess of bitter taste will increase vada or dryness. Excess of starch and sugar will increase kaba or like, let's say, cold-like symptoms, which is just a small portion of the whole thing. And excess of fat, what imbalance it causes? So what is the apt diet which can keep all these in a dynamic equilibrium? That is when everything is not in excess. So what is that food? It is the food which was there in ancient times. Anything which you can eat, which you feel like eating without cooking. So you take banana, you take sprouted groundnut, or you take apple or carrot. All these have low calorie. They do not have increase in starch. So when you don't eat high carb diet, you have a balanced kaba. When you don't have a high bitter food, you have a balanced vada. And when you don't take increased salt, again you have a balanced kaba. So there is a direct relationship between the kind of taste molecules and the imbalance happening in your body. But what is this imbalance? We need a multi-omics personalized medicine based on metabolomics, so mass spectrometry based experiments to clearly identify what food molecule cause what molecules as imbalance. For example, increase in glucose and starch food cause an imbalance in kaba dosha, or it can cause an imbalance in cytokines. So these cytokines can be identified by mass spectrometry and multi-omics personalized medicine. So when you get a patient, it's not that easy for you doctors, because they come to you after failing in all other treatments. So you cannot start with the actual traditional treatment. So you need to identify which stage they are in. So you need a really personalized medicine. All of the patients do not look same. Like some patients have, you will be providing it will be a cancer patient, but they will have a mutation in mTOR pathway. So autophagy is not going to work there. So without knowing that you are giving them fasting, you are not going to cure the disease. So you need to have a clear understanding which signaling pathway is mutated, which kind of imbalance is there in the body, and which inflammatory response or which kind of excessive secretion like mucus secretion, insulin, whatever secretion hormones is abnormal. So we need to have a laboratory to characterize this research. So when we have a patient, we need to characterize the patient's molecular condition, then design a personalized medicine. So this is our dream, and which Dr. Praveen and Dr. Arun is also thinking of. We need to have a common platform to discuss. You are a huge group of like 4,000 people joining in this meeting. We need to collect so much of information to understand this fundamental cause of disease development, then which patient has what type of mutation and how to treat them, what kind of so this will bring very precise type of treatment which can accurately cure the disease. So I will wind up with this, and thank you so much for joining me. It was a wonderful effort from Dr. Praveen and Dr. Arun and all this huge group, and I hope that we will have good discussion in the upcoming years. And I hope that in 10 years from now, we'll have a full-fledged laboratory and we will be able to accurately predict what is the imbalance in the body, and we'll be able to accurately give that specific medicine to cure the disease successfully. So I hope this will open up a window and it will give light for all of you to search for natural truth of the fundamental cause of disease formation and the fundamental light of health. So thank you so much.
Thank you, sir, for the enlightening session, especially all three sessions. Right now, we'll take a few questions pertaining to your last session.
Yeah.
Okay. Ms. Pavitra asks, what are the other mechanisms of activating, sorry, this is for the first session. What are the other mechanisms of activating autophagy other than fasting?
Okay, so now see, this research is funded by pharma. So now we have medicine to inhibit mTOR and you can activate autophagy. Well, that is an artificial method, and the most important way to activate autophagy is fasting. This is the top most powerful method.
Okay.
You don't have a what you say, shortcut.
Shortcut. Okay. So the next question is, in case there's already a chronic degenerative disease that has developed, what are the chances of improvement through fasting and autophagy?
Okay, so this is a very important question. What are the like chronic disease which has developed? And so you are telling that which stage autophagy can work?
Degenerative. Basically, he's talking about the several chronic degenerative diseases. What are the chances of reversal or cure through autophagy and fasting?
Okay, so once the neurons are dead, the problem with the brain is that the neurons are for lifetime. You need to protect them throughout your life. You can have new neurons, but it is not that good repairable like a liver or leg. So once a neuron is dead, there are reports from NIH group Professor Mark Madsen. You can read more about him. He has done enormous work on fasting and brain neurons, how he has shown that new neurons can be formed when you are fasting. And it can actually try to bypass the dead neurons and form a new connection if you simultaneously give stimulation. I haven't read in depth about that, but how can autophagy help here? Autophagy, what it does is, so why a neuron die? Because there is accumulated damaged mitochondria which is causing oxidative stress, and there is accumulated misfolded proteins which is causing neurodegeneration, like in Parkinson's disease or Alzheimer's disease. So if you can remove these proteins, the neurons are still alive, they are going to die. If you can remove these proteins by autophagy or some other mechanism, there are many other mechanisms, like you can just throw out the protein by lysosomal exocytosis. So there is something called chaperone mediated autophagy, where you can specifically bring a protein into the lysosome and throw out. So many things are there. So if you can trigger them by herbal medicine, we do not know. So fasting alone, I would say, is not that clinically reported, or I need to read more. So if you can remove the misfolded proteins and mitochondria, damaged mitochondria, you can rescue that neuron from dying. Then if you rescue the neuron from dying, and if you have a proper stimulation of enriched environment, this enriched environment means a happy environment which has a lot of exercise and stimulation for the neurons, it will develop synaptic connections and improve the memory. So neurons are like muscles. Like if you don't exercise, you lose your muscles. But if you exercise, it stimulates your muscles to grow more, and you will develop into a gigantic structure. Neurons are just like that. Even if neurons are not working properly, if you are stimulating it by signals like exercise, it can generate, out of 1,000 synapses, it will develop millions of, let's say, I don't exaggerate, let's say 10,000 synapses. So it will try to bypass the problem and improve the synaptic signaling. So you can actually recover from this kind of, that's why you can see that physiotherapy and fasting can help to recover the stroke patients to some extent. But anyway, we are a long way to identify how to protect these dying neurons. We need to have really good medicine screening from plants and chemicals to understand this protecting neurons. Okay, sorry.
Okay. And what about the electric stimulation of the neurons?
Yeah, so electric stimulation, this is what I said, it can be recovered if the neuron is alive. What are electric signals? It is passing through the synaptic connections. So even though the synaptic connections broke down, you can have 10,000 more synaptic connections if you are stimulating the neurons. So if neuron is alive, you can rescue. This is the whole point. That's why stroke patients, even though they are paralyzed, you can find that after they are doing exercise for 3 months, they are coming back. Even though they had coma, they were in coma for like 3 months after an accident. Slowly they recover with physiotherapy, because exercise stimulates new 10,000 synapses to form. So what is the difference between a, let's say, Sachin Tendulkar, the cricket master, if you are familiar with, compared to others? He can hit the ball in whatever way it comes. Why his brain is different from us is that the motor neurons controlling this batsman's muscles in the hands have, let's say, millions of more connections than what we have, because of constant training. So neurons are not just neurons. It's the main thing is the synaptic connections. If there are 10 synaptic connections which can be increased to 10,000 synaptic connections, even though one neuron is surviving, it's going to function equivalent to other 100 neurons. So it can take over the function of 100 other neurons, even if side by neurons are dead. Okay.
Yeah. So the next question is, can a high-grade cancer patient be given fasting, and whether autophagy mechanism will work in them?
Yeah, thank you for asking this question. Very important. So autophagy is a double-edged sword. In high stage cancer, you have a huge blob of cancer tissue. So the blood vessels cannot reach the inner core of the cancer tissue. So how do the cells inside the cancer tissue survive? They need nutrition, but the nutrition is not reaching through the blood vessel. Then what they do is, when there is a nutrition deficiency in the inner core of cancer cells upregulate autophagy to survive. So here, autophagy is used to recycle the existing proteins and lipid to stay alive. So cancer cells stay alive by upregulating autophagy. That's why when you get cancer patients in very late stage, they come to you in a very late stage when everything fails. So if you do fasting, you are not going to cure them. So you need a very branched approach from several treatment field. Like, let's say Siddha medicine. They have a very advanced level of treatment compared to the existing allopathic treatments, which use. So you might see that the allopathic treatment use platinum-based tablets, cisplatin. What they do is they will inhibit any dividing cell. But if you look into Siddha medicine, they collect metallic roots, so from this medicinal mountains, and their preparation has very effective arsenic and metal-based ash, which will work much effectively than the cisplatin. So what I'm trying to say is, we are holding only 1 feet of the whole elephant. We need all the knowledge from all the medicines in a well calculated way to treat the advanced cases of diseases. Fasting cannot cure everything. In the advanced cases, the system is already broken down. So you need to see if the fasting based pathway is still functional or not. If mTOR is mutated, you cannot expect autophagy to work. Then you need to provide a drug which can stimulate autophagy. So several herbal medicines are screened and there are several plants which can activate autophagy. They will be eventually coming to pharma companies, but anyways, so you cannot treat everything. This is what we need to understand, the limitations. So only after understanding what stage they are, which pathways are active, this we need to identify by multi-omics personalized medicine, which we are dreaming of a laboratory in future. So you can actually see this cancer patient as which all pathways are active. Then only then you can put that particular pathway into action. So there is apoptosis inducers which can actually kill the cell, cancer cell specifically. I would wish to bring one point here. If you are fasting and you have cancer and you are taking chemotherapy, there's an important work from Professor Walter Longo showing that when you are in a fasting diet for 3 to 4 days and you are doing chemotherapy, the cancer cells will be specifically killed and the normal cells will not die. Stem cells will not die. So the chances of cancer reoccurrence is also less. So this I will cover in the next talk if someday we are meeting again. So this is based on the theory that I told about Warburg shift. Only it doesn't make 36 ATP, only 2 ATPs are made. So cancer cells require 10-fold higher glucose uptake, but you are fasting. So cancer cells cannot divide. It cannot make its raw materials to divide. In addition to that, you are providing a chemotherapy. So cancer cells try to divide, but they will die because of chemotherapy. But the normal cells turn into a quiescent form. They do not divide during fasting. So normal cells are saved and cancer cells specifically die. So a lot of research are coming up combining this, so that I really hope in the coming 10 years we will be able to have a much advanced cure for these advanced stages of cancer and neurodegeneration and liver cirrhosis and diabetes, all these. Yeah, thank you. And
Okay, so the last and the simple question. Generally, since fasting is always accompanied with rest and relaxation as a notion,
yeah,
is it really necessary to reduce or limit our physical activities during fasting, or a person can continue?
So listen to your brain. Whatever brain is telling you is the final command. We do not know anything. Brain knows. So when you are sick, you are tired.
Yes.
So brain wants you to take rest. Brain demands you to take rest. But you know that you will take a paracetamol and you will go for work. So you are really torturing the immune system. I explained this whole talk, how much torture you are going to give to the immune system and the brain. So rest is extremely important to make this happen properly, because let's say you are eating food, okay? Now the blood circulate, there's something we need to understand about energy balancing in the body. So you are eating food, now the blood circulation and oxygen is required for the stomach and the intestine. So you will feel sleepy because not enough blood supply and oxygen for the brain. So of course you are eating a high caloric food. You need more glucose and oxygen for digestion. So there is a diversion of energy to the intestine. If you are running around while eating food, brain need to take care of the muscles. Muscles take a huge amount of energy. So how can brain manage muscle plus intestinal muscles? You are going to create a problem for the brain. So a virus infection is much dangerous than all these. When immune system is at full stretch action, you need huge amount of energy for the immune system. So brain is focusing on the immune system, not on your muscles, on your leg. That is why brain is withdrawing energy from your muscles. You are feeling weak, you are feeling lethargy, you need to take rest. Okay, this is same as you are asking that when there is a enemy invasion in the country, can we continue with our regular routine of entertainments, building or playing cricket and playing sports and our cultural activities and enjoying our food and movie? No, we need to stop everything and prepare for the defense. So same as that, we need to use all the currency available for defense. We cannot use it for whatever the country was doing for growth purpose. Okay, yeah.
Yes, thank you, sir. We understand that conserving our body's resources to fight the infection.
Energy balancing is a very important concept we need to understand. And you should also think that there is something called healing crisis, which means that after a prolonged fasting, body is trying to eliminate the toxin by all the means possible. So it cannot eliminate through kidney will get damaged. It cannot eliminate through liver is getting damaged. It cannot just vomit off. It will try to eliminate through skin. You will apply some suppressive therapy. What it does, it need to store. It is like the Shiva is drinking the poison and it cannot go anywhere, right? So it's same. You have some toxins inside your body and it tries all the way to eliminate it, but you will give suppressive therapy. Then you need to store somewhere. Where it will store? It will store in the most unused portions of your body, which is like prostate, mammary glands, something which won't cause immediate death. Like it won't store in heart muscles, because it will store heart muscles. The heart muscle cell DNA will get mutated. There will be cancer in the heart muscle. you will die immediately, but it won't happen. It will store the toxin in the. So, what is the fundamental cause of cancer? Is the toxins which create mutations in the DNA. It will try to redirect it to mammary gland or prostate gland, or these are just my philosophy. So, brain is working there, trying to eliminate it, and yeah, so this we need to understand from our own inner voice, and how to balance this, how to help and support our inner god-like immune system and brain, trying to save us from all kind of tortures and stress we are going to do in 80 to 100 years. We should remember that our lifespan is 130 years. We are living only half of our lifespan because we have tortured our body so much that we end up just living half of our lifespan. Okay.
Thank you, sir, Dr. Raghuram Swaminathan. It was indeed a very enlightening session where, as a student or a physician or any leading expert in the field, we really needed to rethink on our strategies to treat the patients. And a very simplified version of the explanations that you have given. Why is it important to help our body to detox, so that, especially through fasting, to promote autophagy, and what is the significance of calorie restrictions to control the intracellular metabolic pathways to kind of prevent fatalities, and at last the beautifully explained molecular mechanism at the cellular levels of how the doshas and the composition of the body is very important to treat a disease. So we really thank you for all the time that you have. It was an honor to listen to you, and we definitely will work on your call to develop a personalized treatment approach to understand a individual as he is, and then treat a person on each and every disease.
This is what I feel like so important to develop this treatment, because we need to clearly understand which pathway is not working. Only then we can put efficient herbs specific for that. So in the coming 10 years, we really need to advance in the research wing of this field. And I thank Dr. Rao for all this support and help. And I thank Dr. Praveen for all the hard work he has done to bring all us together. And I thank Dr. Arun. And this is our dream that in the coming 10 years, if we can use Dr. Praveen's website as a platform, we should extensively discuss the clinical protocols and which of the protocols can succeed, and extensively discuss what are the molecular basis of this. About, you need to start reading research papers on this. One of the famous people are like Professor Walter Longo, Professor Mark Madsen, then Professor Roy Wolford. Then a lot of work going on in this field, but you should expose to all these and read a lot, and let us have a common discussion platform and develop the research wing of this group. So we will be able to extensively and very successfully treat advanced diseases. So thank you a lot for everybody who is joining this.
Thank you, sir. Thank you once again.